Okay. All right. Thank you very much. All right. So before I start, I actually want to thank the people that did all the work. So I'm very fortunate to work with some very talented and motivated people. So I particularly want to highlight John here. You can see the cursor, who's a postdoc who really got this project to work. And then there's Oliver and Gabriele, who are also both PhD students, also working on this project. Marcel is a titular professor in my group who's done a bunch of the reconstructions I'm going to show you, and then Nathan Constantine did a bunch of the water work. And I thought I should really thank the Dubochet Center in Lausanne. They've set up these fantastic machines, and you'll see some of the reconstructions we've done on them. All right. So how fast are proteins? So proteins have a bunch of characteristic timescales. So there's some very fast timescales. For example, the motion of side chains, or the motions of the backbone. Those occur typically on a timescale of picoseconds or nanoseconds. Maybe the motions we might be most interested in are actually large amplitude motions that are usually the ones that are associated with the particular task that a protein fulfills. And these large amplitude motions tend to occur on a timescale of microseconds to milliseconds, right? And so now, right, so the question then is, right, so if most of what we know about proteins is dictated by static structures, then we must be missing essential pieces of information. So what we would really like to do is to observe these proteins as they perform a particular task. And what we would need, of course, is a technique that offers both atomic spatial resolution as well as a time resolution of at least microseconds, right? So if you were to ask what is the characteristic time scale associated with the catalytic activity of an enzyme, the answer would be typically microseconds. And so there is time-resolved cryo-EM, right? And so the approach here essentially consists in taking your sample, and then you spray a compound on it or expose your sample to a pulse of light, and then that way you trigger some dynamics, and as they unfold, you rapidly plunge freeze, and you hope to trap some intermediates of your dynamics and then image them later on. And the catch here is that this technique essentially is limited by the time scale that it takes to plunge freeze your sample, and that's on the order of 1 millisecond. So this is way too slow to capture many of the dynamics that we would be interested in. All right, so what if we could do the following experiment? And so this is something we've been working on for a while now. What if we could take our cryo sample, where we now have our protein here embedded in vitreous ice, and so now we come in with a laser beam to very rapidly melt that sample, and so the protein then finds itself somewhere near room temperature in solution, and so now it can conceivably undergo some dynamics that we might trigger with some sort of external stimulus. And as these dynamics unfold, we switch off the laser that keeps everything liquid, and then we allow the sample to recool very rapidly, and we'll vitrify it so that it traps these proteins in their transient configurations, in which we can afterwards image them with standard cryo-EM techniques and get a reconstruction and all of this, right? And so this sounds a little bit like a counterintuitive idea. So for starters, you're not supposed to warm up a cryo sample. Usually if you do this, it crashes out and you have to throw it away. And then the other point is, it's also not so clear if this step here should work, which is, well, assuming here that the sample will cool so rapidly that it will vitrify, and it turns out you need cooling rates in excess of 10 to the 6 Kelvin per second, right? And so it's not even clear if this would work. And so the purpose of this task is I'll show you that this actually does work. So this is how we do the experiments in our lab. So we have a transmission electron microscope that we've modified for time-resolved experiments. And so essentially, we come in with a laser pulse here from the side, and it's bounced off a little mirror, and then it's directed at the sample. Samples we usually use are these ultrafoil grids, where you have holy gold film on top of a gold mesh. And so now you come in with a laser here. It's a green laser that gets absorbed by the gold. And that laser is now focused to a spot size of, say, something like 30 microns, and it very locally melts your cryo sample. It stays liquid for a while, and then you can use this to try and observe dynamics. So the very first thing that we did to show that this would even work was we made a bad cryo sample. So this is essentially a cryo sample of hexagonal ice. And so the diffraction pattern here tells you that this is hexagonal ice. And so if what I'm telling you is correct, then the following should happen. So we shoot a 20 microsecond laser pulse at our sample. If what I'm telling you is correct, the sample should melt. And after we switch off the laser, it should cool so rapidly that it vitrifies. And that's exactly what we see. So after the laser pulse, we see that the contrast here has gotten homogeneous, and the diffraction pattern tells us that this is vitreous ice. So we're evidently able to melt these samples in situ, and then they cool so rapidly that they revitrify. So now the question is, how fast do they actually cool? And so what we did here was an experiment where we used very short electron pulses of single nanosecond duration to actually capture the temperature evolution of the sample while we irradiated with a laser. And so this is what the sample looks like in low magnification. And so now we collect the selected area diffraction pattern here from this little area here that's marked by a dot. And so you just see the polycrystalline gold here. And so now essentially we use the gold as a thermometer to probe the temperature evolution of the sample. So, and now as we come in with a laser beam to heat up the sample, we take single nanosecond electron pulses to capture diffraction patterns as we move along. And so what I'm plotting here is the diffraction intensity, of this gold diffraction pattern as a function of time. And so now what you see is that the moment we switch on the laser, the diffraction intensity drops very rapidly in just 1.2 microseconds. And this essentially tells you that the sample gets warm, right? So this is the Biot-Wald behavior. So the temperature shoots up. And then very fortunately, the temperature stabilizes. Essentially, this you can tell this by the diffraction intensity leveling off here. So temperature of your sample stabilizes. And then the moment you switch over the laser, diffraction intensity recovers. And the time scale here we find is 3.6 microseconds. So that's the time scale on which your sample cools down, right? And so if we simulate this process, we get exactly the same time scales. So this is the red curve here. So this all agrees very well. And so this, these timescales, this is just the natural heating and cooling timescale of these samples. I should add that this experiment here was done in the absence of any ice on the sample. That makes the experiment very hard. But we can add the ice to our sample, of course, in the simulation. And you see this is these bluish, greenish curves. You can see that things get a little bit slower, right? So the ice has some heat capacity that adds inertia to the system, but the timescales remain about the same on the order of a couple of microseconds. All right, so now what you got to realize is that the timescale on which the sample cools down, that is the timescale on which you trap your particles, right? So this is the time resolution of your experiment. So from this here, we can conclude that with this sort of approach, we'd get a time resolution of, say, 5 microseconds or better. And that's about 1,000 times faster than what's been done with traditional approaches. All right, so now in this experiment, we've really probed the temperature of the gold. And what you'd really like to do is you'd like to probe the water. And I'll go over this because this would be a talk of its own. But we also do this. We essentially, while we heat our sample here with a laser, we come in with electron pulse capture a time resolved diffraction pattern of the water, and then we can follow the structural evolution of the water as as we melt it and heat it to to somewhere close to room temperature, and essentially we get the same picture. The timescales are exactly the same. The one thing that's interesting here is we figured out that in the process we make it through most of what is called no man's land and almost like above 0 degrees Celsius without crystallization, and all the way at the end maybe there's a tiny bit of cubic ice that forms, and I'll show you later that that doesn't really matter for our experiment. All right, so can we do this with a protein in there? So the very first thing we did was we did a very simple proof of principle experiment, right? It's very well known that proteins take damage under electron beam irradiation, right? Some of this damage just consists simply in breaking bonds. And naively, you should think that, well, if I break all these bonds, why does my protein not fall apart? And the conventional wisdom here is that this is not the case because the vitreous ice matrix just keeps these fragments in place. However, if we now come in with a laser pulse and we melt our sample, then these fragments should have the ability to just diffuse away. So we should see the protein unravel, and then when we switch over the laser, we should be able to trap these fragments in place and see partially disassembled proteins, right? And this is exactly what we see in our experiment. So here we've illuminated a cryosample of apoferritin with a dose of 5 electrons per square angstrom here in the upper left, and in the bottom right we used 10 electrons per square angstrom, and what you see there is just like normal apoferritins. So now, if this experiment works, then the following should happen. When we melt and revitrify the sample, proteins in these areas here where we have done nothing to them, they should be perfectly happy. They should revitrify and show us pristine apoferritin. However, in these areas that we've beam damaged, we should see these particles unravel. And this is exactly what we observe. So in this area up here in the top right that we haven't irradiated previously, you just see normal apoferritins. And then if you go to this area that we've exposed to a dose of 5 electrons per square angstrom, you see there's heavy fragmentation, and there's barely any particles that are actually still intact. And then this area here that has seen 10 electrons per square angstrom, there's barely anything left. There's just a couple of fragments that barely have any contrast. Right? So essentially, this shows that we can melt these samples in situ. Once they're liquid, they allow particle dynamics to occur, and then we can revitrify the sample and trap the particles in whatever state they're in at that point in time. All right, so the next question, of course, is these particles that I'm telling you that I'm telling you they're fine, but how do we actually know? And of course, we have to do a reconstruction here to prove that these particles are okay. And so we did an experiment here on CCMV, which is an icosahedral virus, and this is on our microscope. The resolution we get is something like this. So this is what a reconstruction looks like on an ordinary cryo sample. Now we did the following experiment where we just melt this sample, keep it liquid for a while, and then let it revitrify, and we do another reconstruction at the end. And the question now is the reconstruction going to look the same? And that's what we find. The reconstruction looks within the resolution that we have. The sample is identical. So our process doesn't seem to do any damage to these particles at all. Here's the same result for apoferidin, where our resolution got a little bit better. And again, within the resolution that we have, there really is no difference. What we've done recently then is we've taken these samples out of our instrument. So we melted and revertified them on our instrument, and then transferred them to the Dubochet Center, where we can now, we can repeat the same thing and now image with the Titan Krios. And so this is what we get. So on the conventional sample, we get 1.6 angstroms, and the revertified one is very similar with 1.5. And there's no difference between these particles. We've tried to look for one. We haven't found any. And this goes down to like the side chain densities, the positions of waters, and so on. And so for all means and purposes, these say within the resolution that we have, 1.6 or 1.5 angstroms, these samples are identical. All right, let me just quickly check the time. Okay, so as soon as we had this and we said, okay, we should also look at what the beam-induced specific motion does in these samples here, right? The difference between a standard cryo sample and these revitrified samples is that the revitrification, the cooling rate is about 1 to 2 orders of magnitude higher. And my intuition would have been that we build up much more stress in these samples just because we cool so much faster, and that this should lead to much more drift, and it's not what we see. So plotted here is the cumulative drift for apoferidin and CCMV for both the conventional samples here in light gray, and then the ones that we revitrified in this purplish color. And you see that the magnitude of drift is about the same. The revitrified samples drift a little bit more rapidly in the beginning, and then they drift more slowly in the end. So overall, the magnitude of the drift is about the same, fortunately. However, what's maybe interesting here is that one seems to be able to influence or affect the kind of drift and the kind of specimen movement that you see. And so this maybe opens perspective to, in the future, use lasers to eliminate some specimen drift on conventional samples. All right. I wanted to say 2 words about how you control temperature in these samples. Right, so, or maybe the question is even more basic. So, you shoot a laser at this sample, and it's vitreous before and it's vitreous after. So how do you even know that you've done anything to this sample? So this is a micrograph here taken after we've exposed the sample to a laser pulse. The green dot here, this is about the size of the laser spot in the sample plane. And so now if we zoom in here, and look at these individual holes, then you'll see the following, and that's very characteristic for these experiments that we're doing. So far away from the center of the laser focus, somewhere out here, we see that nothing has happened to the sample, so we just see vitreous ice here. And then as we move further in, we encounter these holes here where we see a bunch of cubic ice crystals. So those are areas of the sample where the sample has just warmed up barely, and so it has just devitrified, and that's it. And then further in, we get samples that contain more and more hexagonal ice and larger crystals. And then in the center, near the center of the laser focus, then we get these holes here where the sample has melted and has revitrified, and these samples send infection pattern tells us this is vitreous ice. Right? And so now, if we simulate the temperature evolution of this experiment, then essentially we can identify this area here, this outline here of the revitrified area with a 0 degrees Celsius isotherm. right? So essentially meaning the areas that revitrified are the ones that go above 0 degrees Celsius. And so now this is very convenient because essentially you can now, after the fact, assess whether your experiment worked or not, and whether the temperature evolution in your sample is the same between different areas, right? So if you don't see the sort of pattern where you have vitreous ice on the outside and then a ring of crystalline areas surrounding this revitrified spot, then you know that your experiment hasn't worked, and you just discard this experiment. Or say if the diameter here of this revitrified area is different, then you know your sample has undergone a different temperature evolution, and so you can also discard this sample. What we've also learned through simulations is that evaporation of these samples plays an important role that can actually be beneficial for the experiment. So that here we're plotting the peak temperature that the sample reaches in I think it's a 30 microsecond laser pulse as a function of laser power. And you see that initially the temperature goes up linearly, and then at some point it starts to level off. And so what happens here is that you hit a temperature at which you start to evaporate your sample, so you get evaporative cooling, and that provides a negative feedback. So at this point here, your sample temperature really doesn't go up any further. And that's very neat to control temperature. If you change the sample geometry, so for example, here we've gone to smaller holes with the same period, then you level off at a different temperature. And if you cap one side of the sample with graphene to prevent evaporation from that side entirely, then you can reach higher temperatures. And this could, for example, be very useful to do temperature jump experiments if that's what you want to do. All right. Okay. The one thing I haven't really talked about is what the stimulus here is, right? So we can melt these samples, and so now we have our protein in solution, and we can hope for some dynamics to happen. But how would you actually trigger those dynamics? And there's a number of ways I can think about how you might want to do this. So one way would be to just use another laser pulse. So if you have a photoactive protein, you just pump that system and you trigger some photophysics in your sample, and you could drive this process. And of course, not all proteins are photoactive. But the other thing you could do very easily is a temperature jump experiment. You just turn up your laser power and your protein will get hot, and then you could, for example, look at the temperature jump induced unfolding of a protein. But maybe the most general way to drive these dynamics, and what I think might be the most powerful way of doing this, is to use photorelease compounds. And so chemical biologists have figured out all of these compounds that are light activated and that allow you to release reagents, right? So in the most simple case, you could take a photoacid or a photobase, and you irradiate a compound like this, it releases protons or hydroxide ions, and you get a temperature jump, and you can drive dynamics of the temperature jump. There's caged ATP, so you can uncage ATP just with light. There's caged ions, calcium, for example, like caged peptides, and so on. And now, I think this would be the easiest way to drive these experiments, to drive dynamics in these experiments. And I'll give you one example of where we've attempted to do this. All right. So the idea here is, could we do a pH jump experiment on a protein and see what happens, right? And so this is a very classical experiment, right? You use a pH jump and you watch your protein unfold, right? And you might want to do this to just look at protein folding, just ask some very general questions. And the way the experiment would work is like this. So you plunge freeze your sample here at pH 7 in the presence of a photoacid. And so now, even before you do anything to your sample, before you load it into your microscope, you irradiate this frozen sample here and you release this photoacid. And so by doing so, you drop your pH, and we figured out that we can drop the pH very low. We can easily go below 4. Maybe it can go to like 2.5 or something like this. Depends on how much light you're willing to put on your sample and so on. And so now the protein that is sitting in your vitreous ice here sees a massive pH change. However, there's no way it can react to it because it's stuck in the matrix of the vitreous ice. However, the moment you melt your sample, this pH jump can become active and the protein can react to it. And then you should see the protein unfold. And when you switch off the laser, you should be able to revitrify your sample and trap the protein somewhere on early timescales to see how this unfolding happens. Right, and so this could be a very general feature for these sorts of experiments, where you take a caged compound, you release it in the plunge frozen state, and then the compound only becomes active when you melt the sample. All right, so this is an apoferritin sample where we have added a photoacid and we've irradiated the sample, and so the pH has gone quite low. And how low it's gone, we'll have to characterize exactly. But before we do anything else with the sample, those are just normal and happy apoferidins, right? Now if we melt a sample like this and revitrify it, this is what we get. And now you can, just by looking at this, you can tell that these apoferidins are not happy anymore. They don't look very good. I should add the following here. If we just melt and revitrify a sample without photoacid, the images we get afterwards, they look identical. If we have photoacid in there, but we don't release it, the images afterwards also look identical. The particles look identical. There's no change. If we release some of the photoacid, but not a large amount, we also see no change. And these are sorts of images you only get if you release a large amount of photoacid. And of course, now what we've done is so I'll have to apologize because the data are only a couple of days old, but we've done some very preliminary reconstructions here. And so this is now a cross-section through our apoferidin of this sample here before we fit it with the laser. So this sample is already at very low pH. Photoacid is released, but we haven't melted and revitrified yet, and you just see like a normal apoferidin. And if you do a reconstruction of the apoferidin afterwards, you see a couple of changes. So the first one that is very obvious is that the amino acids here, the surface of the protein, they seem to lose quite some resolution. And so it's obvious that this protein starts to disorder as it's exposed to the pH jump. And then if you look at different amino acids there, you see a couple of changes. There are some parts where you lose density, and those seem to be regions that disorder, and so they disappear from this reconstruction. And then there are other parts of the protein where you see very distinct changes. And so we're at the first stages of just analyzing this, but I'll show you one of these changes that we've seen. And so this here is in the sample before we melt and revitrify. This is an interaction here between an aspartate and an arginine. And so these two here form a sort of a double hydrogen bond. And if we melt and revitrify the sample, we get something that looks like this, right? So the model we fitted in here, this is just the molecular model of pristine apoferidin. And so you see that this model, after we melt and revitrify, doesn't fit anymore, right? So, these densities don't seem to touch really anymore. And now you have to be a little bit careful how you interpret these densities. This sort of change we don't see if we just revitrify the sample without photoacid. We also don't see this sort of change if we don't revitrify the photoacid. And we also don't see this sort of change if we don't release enough of the photoacid. What seems to have happened here, and this sort of makes me a little bit optimistic that this is actually real, is that at the pH that we're likely reaching, which must be 4-ish or lower, you'll very likely protonate this aspartate. And by doing so, you'll break up this hydrogen bond, and I believe that this is what we've observed here. And then we have to go through this. One thing we obviously have to do is we have to do some sort of conformational sorting here, which we haven't done. You can see that these data just came in. There's also a lot of these particles don't look very good, and I think right now we're just throwing them out, and there's probably a lot to learn which we haven't done yet. All right. I hope I didn't go too much over. So I'll just quickly sum up to you what I've told you about. So this is, I've talked to you about the concept for doing microsecond time resolved cryo-EM, where the idea essentially is that you take a normal cryo sample, you just very rapidly melt it with a laser, you keep it liquid, which provides you with a time window where you can allow dynamics to occur, trigger them with some sort of stimulus, and then as you switch off the laser, that keeps everything liquid. The sample cools very rapidly, the sample revitrifies, and you trap particles in transient states in which you can image them afterwards. So I've shown you that you can actually do this. You can melt and revitrify these cryo samples, which we've done here with by just showing you that you can, turn a hexagonal ice sample into a vitreous ice sample. We've also characterized the time resolution that this technique has. And we're finding something of 5 microseconds or better. So just by changing the sample geometry and changing the heat transfer problem, we think we should easily be able to go down to like maybe 1 microsecond. Maybe what I haven't really answered is, why is this actually so fast? Well, the reason is that you melt your sample only very, very locally. You put a small amount of heat in a very small area. And the moment you switch off the laser, this hotspot finds itself surrounded by the rest of the sample, which has a very large heat capacity and is still at cryogenic temperature. And this is how you get these fast cooling timescales. Showing you a proof of principle experiment where we beam damage some particles, and then once we melt the sample, the fragments just diffuse apart, and we can then trap particles in these partially disassembled states. I've shown you that this process is very benign to the structure of the particle. So we've been able to get high resolution reconstructions here where we find no difference at all between a conventional cryosample and a cryosample that we've melted and revitrified, showing that the process leaves the protein structure intact. And maybe if you think about why that is, well, the protein does not absorb the laser, right? The only thing that absorbs the laser is your gold support, and then the gold warms the ice, and the ice warms the protein. So the laser really doesn't have any way of damaging your protein. And then in terms of what you're doing to the protein, is you're really not doing anything to it that you haven't done to it already, right? I mean, you've already vitrified it, and now you're just taking the protein through the same process twice more, like once forward and once in reverse. And so the protein seems to be perfectly happy to do this. And then finally, I told you a little bit about very initial experiments on doing pH jumps on these samples where, right, this is essentially one example that we're trying to do here as part of a broader concept, where the idea would be to use caged compounds that you embed in your cryosample, and then you release these caged compounds before you do the melting and vitrification, and the compound that you release only becomes active once your sample is liquid. And so I believe this should be quite a general approach to drive quite a range of dynamics in your samples. And that's what I had. So thank you very much for your attention, and I'll be happy to take questions.